Mr. Quake nel suo laboratorio si occupa di DNA circolante, microbioma e trapianti d'organo. Che relazione c'è tra questi tre elementi?
A part of my research involves the study of DNA that circulates in the blood. These DNA molecules are not part of the nucleus of cells anymore, but they're kind of the results of cells that die and break open and dump their genetic material into the blood. The DNA gets chewed up into small pieces, and most of this DNA in your blood is human. And we've, over the years, used that as a way to develop diagnostics for prenatal genetic health, to make a replacement for amniocentesis, as a test for Down syndrome and other aneuploidies. We've also used it as a method for measuring rejection in organ transplant recipients. After you've had a new heart transplant or lung transplant, we've developed a blood test that allows the doctor to know if the body is having a rejection event from the organ. And we've also discovered that some of that DNA is from your microbiome, and it provides a very powerful global view into your microbiome, because pretty much blood gets into every part of your body, and so wherever there are microorganisms in your body, they contribute to the cell-free DNA in the blood. And about 1% of the DNA in the blood is non-human and from your microbiome. So we sequence that DNA and look to see what's represented in your body. With our organ transplant cohort, we had a unique opportunity to ask how the microbiome changes as your immune system gets turned off, because all of those patients are taking strong immunosuppressant drugs, and so we were able to follow how their microbiomes changed as their pharmaceutical suppression of the immune system was increased over time. And it's a very interesting story. We discovered which viruses were in the most intimate dance with the immune system, and that it was dominated by a viral response and not a bacterial response, who seemed to be much less sensitive to perturbations of the immune system. And that was a very interesting result that we stumbled upon, almost by accident, without planning it, because we had taken the data for studies of rejection, not for microbiome analysis. But that's sometimes how science works. You get into a new area by a fluke observation, and it's been very rewarding for us to enter the field.